And be sure that is recording. Okay. Okay, good afternoon, everyone. Welcome to the CCMAR webinar, today with Alicia Bruzos. Alicia is a biologist and holds a master's degree in bioinformatics, and currently she is a doctoral candidate at the Center for Research in Molecular Medicine and Chronic Disease in Santiago Compostela. The topic of today will be about the clam species in the seas of Southern Europe, and these species are usually have again tendency to develop cancer and we know that since the 60s. And today Alicia will tell us how mitochondrial genome sequence of marine leukemias reveal cancer contagion between clam species. Alicia, again, thank you very much for accepting the invitation, and please feel free to share your slides and start your presentation.
Thank you very much, Joao. And thank you very much also for, to the CCMAR for inviting me to do this talk. So can you see this?
Yes, yes, okay. Perfect.
Yeah, so thank you very much. Today we will talk about contagious cancers, as Joao has said before. And more specifically, as we have a big project about this, I will focus on one little project that we have about sequencing some clams, and let's see what happened with them. So first of all, I am here doing this seminar, but the idea was that you had Sarah Rocha doing this seminar, but she has been a mother recently and she couldn't join this date. So yes, as I told before, I will be presenting a little part of the project. So in the future, you could still invite her to tell you the whole story of this project. So yeah, you have that option for the future. So let's go to the topic that we will talk about today. So let's start talking about cancer. Cancer is something that we all know about that, but let's see exactly the definition of cancer. So cancer usually starts with one cell or several that mutate. And those cells start to divide uncontrolled, and they form a mass which will be called primary tumor. Eventually, one of those cells will acquire the ability to abandon the primary tumor where it was originated and conquer other parts of the body. This is the process that we know as metastasis. Metastasis is nowadays the second cause of death worldwide because it's the main cause of death for cancer patients. So out of 6 deaths are caused by this process. Also, metastasis is a really important thing nowadays in cancer because, as you already know probably, it is very important that if you have a cancer, they find it very soon in the early stage, because if they find it in the end stage, you will probably have metastasis, and we don't have treatments, or we don't have many treatments for metastasis. So metastasis is estimated to be the responsible of the 90% of cancer deaths. So as you can imagine, for the health system, it's very important to study this process of metastasis. And here is where we frame our project. But I am giving a seminar in the CCMAR, and the CCMAR is an institute working with sea science. So why am I here? You will know about this later. So yeah, we were talking about cancer and metastasis. Another fact is that if you die, if you have a cancer and you have metastasis, if you die, the cancer will die with you. In the contrary, if that cancer would be able to propagate to other individuals, even if you die, the cancer will be still alive. And this seems like science fiction, but it is not. Actually, in nature, transmissible cancers or contagious cancers exist. Let's talk about them very briefly. So one of them is affecting the Tasmanian devils, which are these animals that you can see in the screen. So Tasmanian devils nowadays, they live in the island of Tasmania. And it's this nice animal that you can see here. And let's see in this video how they behave. So as you can see, they are not very friendly between them. They usually bite each other, and that's a natural behavior of Tasmanian devils. And that's how they can transmit the cancer. So this is the cancer that is transmissible in Tasmanian devils. It is called the FTD. It was firstly recorded in 1996 in that picture that you can see in the top. And in 2007, it was almost covering all the island. So it was propagating very fast. This, as you can imagine, it's a threat for this species. Let's talk about more contagious cancers. Do you like dogs? Well, dogs can also suffer of a contagious cancer. In this case, it's a venereal tumor, so that means that it's transmitted between by coitus, and the tumor, it is located in the genital area. So they have studied this contagious cancer in dogs, and as you can see, it is also spreading all among the world, with more prevalence in countries that are less developed. And finally, let's go to the sea. Let's see why I am talking here in the CCMAR today. So the last, nowadays, the last contagious cancer that has been described is the disseminated or hemic neoplasia. It's a leukemia-like cancer, so it is affecting the cells that are in the hemolymph. The hemolymph is, well, I guess you will know, but the hemolymph is the blood of vivals. And this cancer, it is found in the blood of these animals. So the first, so we know hemic neoplasia or disseminated neoplasia since the 60s, but we didn't know the etiology of the disease till 2015, that it was discovered that horizontal transmission, that means contagion, of this cancer was happening in the sea. And it was discovered with this species that you can see in the slide, Mia in the United States. 1 year later, and several studies more later, they discovered that this contagious cancer affecting vivals was not affecting only that clam, but also those other species like cockles, Terastoderma medullae, that you have here and you have in Algarve as well. Mytilus edulis, Mytilus chilensis, Mytilus trossulus, all these species of mussels, and clams, Mia arenaria, which was the first known case, and Polytitapes aureus as well. So today, I will talk about another species that we didn't know that it has cancer before, and we didn't know that it was contagious cancer. So I will talk about the warty venous clam, this clam that you can see in the picture below. And there you can see how we take the hemolymph, that is the blood of these animals, that as you can see, it's transparent. That's not water, that's actually hemolymph. And we put that hemolymph in a microscope slide, and by cytocentrifugation, we can see the cells. And as you can see here, sometimes those cells, instead of being healthy cells, they have this shape, they are very populated. This is a cancer, a leukemia cancer of this warty venous clam. We have currently this study under reviewing in live, but if you are interested on it, you can download it from the preprint bArchive server. So let's continue talking about this. So, okay, we found a new cancer. We described them. We did some electronic microscopy, and we made the karyotype. We describe it very well. But is it contagious? Let's see. What do we found? So first of all, what we did is to collect several samples, so around 350 samples from 9 different sampling points. So you can see we also have one point in Lisbon. And we found cancer in 2 places. One in the Atlantic Ocean, in 2 points of Galicia, and another one in the Mediterranean Sea, in the Balearic Islands. That was surprising for us. Then we decided to do some Sanger sequencing of some mitochondria fragment. And when we did that, we realized that both cancers, so we have here 2 samples of cancer, one from the Mediterranean and one from the Atlantic Ocean. And as you can see, they are the same. They are exactly more or less the same profile. That was weird. So this cancer might have the same origin. So that was already pointing in the direction that it might be contagious, because if it is contagious, the origin was one single origin some time ago, and the genome will be still the same. So we continue analyzing this, and we sequenced healthy animals from the warty venous clam. That was the animal that was having the cancer. And we realized, okay, we have this profile, but if you look at the places where we have the black arrows, some peak correspond with the warty venous clam, but we have some other peaks that do not correspond with the warty venous clam. So by looking in the genetic databases, we realized that if we took the striped venous clam, which is a different clam but cohabits with the warty venous clam, those second peaks correspond with the peaks of the striped venous clam. So we were, okay, what is happening here? And we continue working on this. So we sequenced by Illumina tumor tissues that will have tumor cells and also healthy cells, because as it is a leukemia-like cancer, when you take the blood and you sequence that hemolymph, you will find tumor and healthy cells. And also, we sequenced a healthy tissue, so a tissue that didn't have a lot of hemolymph, right? So in that tissue, we will have mainly normal, healthy cells, right? Okay, we started aligning the normal tissue against the 2 reference genomes, the 2 mitochondrial reference genome of those 2 species. And as you can see here, the majority of reads of sequences were aligning to the warty venous clam, so the one in the left that has around 4,000 reads there, right? While in the other clam, there was almost nothing, okay? Okay, that was as we expected. When we did this, but with the tumor tissue that will have tumor cells and also some normal cells, we found this. That was also very surprising, because we found that actually for the tumor tissue, the majority of sequences that were there were aligning to the striped venous clam, so the one in the right here, around 2,000 of coverage, while just a little bit were aligning to the warty venous clam. And as I told you before, as we were sequencing hemolymph that have the majority of tumor cells, but some healthy cells, those ones will be these ones that we can find here around 800 coverage of tumor in warty venous clam. So here we were already more or less aware of this story, but we continue working. And what we did there, then we wanted to do some phylogenetic analysis, both with the mitochondria and the nuclear genome. So before dipping into the results, let's think how contagious and non-contagious or normal cancers will behave in a phylogeny. So let's start with the easy case. Let's talk about the non-contagious cancer, so a normal cancer, a human cancer that we usually have. So if we have 3 samples, so 3 patients in this case, and we sequence the tumor and the normals and the no tumor or healthy normal cells, we will get 2 sequence, and we will get that for all the patients. Once we build the phylogeny, the tumor and the normal sequences of one patient will cluster together in the phylogeny because the tumor originated in that patient. So we will find like the 3 patients, the tumor and the normals clustering together. But what if the cancer is contagious? That will be a different case. If the cancer is contagious, the common origin of the tumor cells among all the samples or cocktails or vivals in this case will be a common origin. So they will cluster together. So we will find all those tumor cells clustering together in one part of the phylogeny. And we will find the rest of the healthy tissues clustering in other parts of the phylogeny, right? Okay, so bearing this in mind, allow us to go to our results. So here we have the phylogeny, but I think it's better to use to see this one that is summarized. And as you can see here, when we cluster the sequence of the tumor tissues that will have mainly tumor cells, they will cluster with the strip venous. But those cancer cells, those tumor cells were found in the warty venous clam that is quite far in our phylogeny. So as you can imagine, here what we found is a cancer contagion between 2 clams species. So probably the tumor arose in the strip venous clam some time ago, and now it is spreading among the warty venous clams. And it is spreading in the Mediterranean and the Atlantic coast of Europe. So just to finish a little bit and to catch up all this story that I have been showing you, I have here a little video, and then we can close the seminar.
Marine waters from our seas and oceans are full of minute organisms, such as fungi, algae, bacteria, and cancer cells. Yes, you heard it right. In some cancers, tumor cells behave as parasites that can be directly transmitted between individuals. Despite cancer contagion being a rare event, transmissible cancers seem to be common under the sea. Marine bivalves such as clams, cockles, and mussels can transmit a type of blood cancer called hemic neoplasia, which is characterized by the uncontrolled proliferation of blood-like cells. Eventually, these cancer cells can leave the host that spawned them and live in the marine environment until they infect a new host, spreading across bivalve populations. The European-funded project Scuba Cancers investigated the prevalence of hemic neoplasia in the warty venus, a saltwater clam found in the Atlantic coast of Europe and the Mediterranean Sea. we collected and examined more than 400 clam specimens from 8 sampling points across 5 countries in the seas of Europe, including Spain, Portugal, France, Ireland, and Croatia. This screening found a number of clam specimens infected with this cancer in 2 regions from Spain, one in Galicia in the Atlantic Ocean coast, and another in the Balearic Islands, bathed by the Mediterranean Sea. We sequenced and partially reconstructed the genomes from the tumors, which revealed the presence of DNA sequences from 2 different species. This unexpected finding suggested cross-species transmission of cancer in the warty venus from a second unknown species. Thus, what was the cancer donor species name? Consultation of genetic databases and further genomic analyses determined that tumor cells belonged to a clam species called striped venus, which cohabits with the warty venus in the seas of Europe. Our findings confirm that marine transmissible cancers can jump between species, representing a potential threat for the ecology of the marine environment, and point out the necessity of their identification and genetic characterization for their monitoring and prevention.
Okay, so that was for the conclusions. But before ending this talk, I started to talking about the importance of understanding metastasis and researching about that. But we realized that it is not only important for understanding the process of metastasis, which at the end, a contagious cancer is like a big or large scale metastasis, but also it is important to trace the cancer in our oceans, because
Marine waters from
as you can see, we have nowadays the same cancer affecting clams in the Mediterranean Sea and also the Atlantic coast, the Atlantic Ocean. So moving the animals and all these kind of transports could be a danger for our ecology of the seas. So yeah, it's our ocean. So it's not only to understand better the metastases, but also to do better policies to avoid this a pandemic in our oceans, right? So thank you very much for all your attention. And also this is my group where I work, and all these institutions that have funded the project and my PhD fellowship and so on. So thank you very much. And if you have any questions, I will be willing to answer them.
Thank you very much, Alicia. It was a very interesting and very clear presentation. I'm enjoying a lot. And yes, now people feel free to ask questions in the chat or in voice, since we can manage easily.
I think there is someone with the
yes. So Sergio Fernandez, please feel free to talk, please.
Yeah, thank you so much, Joao. I have just a question to Alicia. I'm sorry not to watching you because my webcam is not working at this moment. I don't know why. Sorry. But was a really nice presentation. Was really interesting to know that the cancer spread from one species to another. But in this case, regarding the Atlantic coast, I know that Camellia gallina is not a very common species here in the Atlantic coast. So what do you think, how this cancer comes to Galicia? Maybe it was another species that brings this cancer to Galicia, or there was some importation of Camellia gallina from Mediterranean coast to the Atlantic coast?
That's actually a really intelligent question. And yes, we also thought on that when we were researching about this topic. And yes, probably the most possible hypothesis is that as in Spain, we are moving animals from from Galicia to Menorca to Balearic Islands, and so on. Maybe some human transportation, so human human touch on this could be the cause. But to know that, I think we need more to study more in detail what happened here, because, for instance, we don't know if this cancer is still propagating, spreading among the Camellia gallina, the striped Venus. So we know that the cancer that is affecting a Vartivinus clam originated in Camellia gallina, but does Camellia gallina still suffer this cancer, or they develop some defenses to not suffer this? So yes, we don't really know at what time, in which moment this cancer arose. So was it in the 90s, or was it in 3,000 years ago? We don't know that either. So before saying, okay, the reason why the cancer is in the Atlantic coast and also the Mediterranean coast is because of the humans, I think it is more important to know the history, the evolutionary history of this cancer, for then knowing it was our fault. That, I guess, it would be probably because the Vartivinus clam in Galicia, we have them, but we don't eat them. They collect it here and they send it to the Balearic Islands, because there it is really the tourists buy a lot of Vartivinus clam there. So yeah, maybe one of those trips, which is the opposite that the one that you were proposing, but maybe something regarding the trade of these animals could be the reason. But yeah.
Thank you so much. I agree with you, Alicia. But even maybe when we import clams from Mediterranean coast to the Atlantic coast, used to be some commercial species like cockles or ruditapes clams. So maybe there are another intermediate host, like or even another species having the same cancer that starts in Camellia gallina, but maybe now it's spreading to another kind of species, because it's not typical for us here in the Atlantic coast to eat Camellia gallina or the I don't remember the name of this clam, but it's not, we don't used to eat them.
Yeah, Venus verrucosa is the other one, the Vartivinus clam. Yeah, I agree with you. We don't know. Actually, this is a very new field, as you could see, and that's why I put the papers with the years. This was discovered in 2015, and then in the next years we discovered it in many other bivalves, but it's quite a recent field. So we don't have a lot of information to know intermediate species. That could be, actually, it's a good hypothesis.
I'm completely sure about it, that if you're looking in some other species, probably you'll find them. So, well, thank you for your answer. It was a really nice presentation. I love it. Thank you, Alicia.
Thank you, Ceci.
Sorry to not see you because my webcam, sorry.
Okay. So while we wait for another question, I have a quick one. So Alicia, the impact of this cancer, what is the impact of these cancers in the clams for the population? If they are really affected or not?
So, the prevalence is not very high nowadays. Well, it's high comparing with cancer. I mean, if that's humans, we will be like, oh my God, what do we have here? Because it's around 10% or so. So yeah, that's not the main cause of death for bivalves in general. But this cancer starts in the hemolymph, but it starts to populate all the hemolymph, the circulatory system of the animals. And in the end, it is all the animal has those cells. For instance, they cannot develop the gonad, so they cannot reproduce. So in the end, in the latest stage of cancer, the animal cannot do anything and it ends up dying. So yeah, it's a cause of death, even though in a commercial scale, it's not that important. There are other parasites that are more
Okay.
important for the agriculture, for instance. But this is a problem, a disease for the animals. So in my opinion, the importance behind this is not that they kill a lot of animals, but that they kill animals and they can spread. So as it happened here, if we are moving animals from one place to another, we think it's not a bad thing, but we are also moving that contagious cancer. And in the end, if we put that cancer in a population that maybe it's more, will die more about this cancer, we can end up with some populations. So it's a real threat for the ecology of these animals. But as for the politicians, we cannot, I mean, the threat of the ecology is not a big issue in the agenda. We can focus in the part that this cancer is killing animals and it is being transported from one place to another. So it's important to trace them and to make policies to avoid humans moving these cancers. So that's like the main importance. And then doing basic research could be very useful for understanding better what happens there, what is metastasis, how does it happen, and yeah, basic research in them.
Yeah. Well, I think that should be a big concern now for aquaculture, mainly that, for instance, now in Algarve is growing companies that produce bivalves from aquaculture. So this probably is another concern that they may need to have in the future. Maybe they already have, I have no idea, just an opinion.
Usually with bivalves, I don't know in Portugal, but in Galicia, I know that they buy seed, like if it was a plant, they buy the seed, the little clams from France, and they plant them in our beaches.
Mm-hmm.
So even though you are bringing seed, so little clams, maybe there are some cancer cells there that are from another species or so, but you are bringing also that here, and you will be infecting your populations, your natural populations from here with a disease that is from other part of the world. So yeah, I think it's very important for overall for the aquaculture enterprises, because it could end up with like COVID,
Yes.
I don't know, like a pandemic in bivalves. And yeah, usually they won't have a vaccine as soon as we have it for humans. So we could end up with the species.
Yes. Well, okay. So do we have more questions?
Yes.
Sure.
Hi, Alicia.
Please.
Thank you for your presentation. It was very good. I have a question about the sampling. When you were doing the sampling, do you collect other species of clams or mussels and analyze them to see if the results were similar or something like that?
Yeah, that's also a good question. So this is a little small part that we weren't planning to do in the scuba cancer project. So yes, when we go on sampling, we usually collect cockles, Terastoder medullae, because it's like our main species of study. We have also collected other species like some other clams, also some mussels, several species and in many points. But yeah, for this species, that's why we don't have a huge number. We only have around 350 specimens, while from cockles we have 6,000. So yes, we collect other species and we at least the ones that we have sequenced, that we have several, we have compared them and no, the cancer of cockles, for instance, is a different one, nothing related with this one. This one was something that we weren't expect. And when we found it, we didn't know that these species have cancer when we started. And we found the cancer and then we started to dive a little bit, okay, what is happening here? And that made us collect also the Camellia gallina, that we weren't collecting it, but when we found, okay, it might be a cancer originated there, we have to collect Camellias gallina. So yeah, it was, as it was not planned, it was unexpected result. We were sampling like while we were working, so it's not the best sampling design, but I think we have covered more or less the whole distribution range of the species, at least in Europe. And we have several points, some of them more distant and some other less, to get an idea of what is happening there. But yeah, it's surprising that in Portugal and Ireland there is no cancer, in Galicia, yes. It's like, yeah, I wouldn't expect that. So that's another fact pointing us in the direction of human moving the animals instead of natural transmission of cancer from the Mediterranean Sea to Galicia, because if that was the case in one direction or the other, you would expect that Portugal will also have this cancer. Yeah. But again, this is a low prevalence disease, so maybe we are not collecting enough animals to detect it in Portugal. So maybe collecting more animals will be a good thing to see if it is really there or not. We are just describing it. I mean, if we really wanted to know in detail what is happening there, we will need more money and more work to do here. But yeah, that's, I don't know if I have answered more or less.
Yes, yes. Thank you.
Thank you for your question.
Do we have more questions? Okay. Let me see. Okay, so we have some congrats in the chat for you, Alicia.
Oh, thank you.
Okay, so if you have no more questions, I guess we can finish our seminar of today. Again, Alicia, thank you very much. It was a great pleasure to have you here today. And please, for all the audience, don't lose next Tuesday another webinar CCMAR, and we still have a schedule for 2 more this year before we close for holidays. And thank you very much for all. Alicia, again, thank you, and see you. Hope you see you soon somewhere.
Yes, thank you. Thank you very much for giving me the opportunity to share with you this research.
Of course. Okay, thank you. Bye bye all. See you next week.